Good afternoon, everyone, and thank you very much to the organizers for inviting me to talk on the clinical development of vaccines. And many thanks to the chairpersons. They have been guiding us in all our clinical development activities, especially Dr. Swati during the Corbevax days. We worked on the COVID vaccines. So, from where Dr. Prabhakar Reddy talked about the regulations controlling clinical trials. So I'll be talking more about clinical development, clinical trials, because the audience is mixed. I try to sort of simplify my presentation, but I'll be open to questions once I'm done with the presentation. So I think I'll just start with a bit of history of how vaccines and vaccines have come. Edward Jenner in 1796 has defined the term vaccine. So what Edward Jenner noticed is those days smallpox was a very life-threatening disease, and what Edward Jenner noticed is the milkmaids who are working with the cattle sheds who are exposed to cowpox and have symptoms of cowpox who never got smallpox. So he took that as an example, and he inoculated a child with cowpox vaccine who developed certain mild symptoms, and then he exposed this child to smallpox, and fortunately the child didn't get any symptoms of smallpox. That's long before all the clinical trial rules and regulations were there. No consent, it is just a scientific experiment those days. So from there we leap into what we are now, where we are with clinical development and vaccine development. So I think you're all aware, thanks to COVID, I think a lot of people got to know what is a phase 1, phase 2, phase 3. I'll dwell upon that in the coming slides. But before we take this phase 1, phase 2, phase 3, which essentially is the clinical development, the most important thing is the preclinical development. So the objectives of preclinical testing is to find the toxicity and the safety parameters of the vaccine, the potency of the vaccine, the immunogenicity of vaccine, and the dose selection. What is the kind of dose they have to take to the phase 1 is determined based on animal studies, and what kind of adjuvants and additives should be added, and how safe they are and how immunogenic they are determined in the preclinical testing. And the animal, there are various models used, and widely used are the non-rodent models, and New Zealand white rabbits are there, and non-human primates like rhesus macaques. And then you have rodent models like Swiss albino and Spragley dolly rats. So after preclinical testing, we move to clinical development. So what is the purpose? What is the objective of the clinical development? First and foremost is to assess the safety of the vaccine. So safety comes first, followed by identifying the appropriate dose of vaccine that is required to elucidate the immunogenic response, and also to assess how good is the immunogenic response of the vaccine. The various phases of clinical trials, we monitor safety to see that there's a continued safety is evaluated in a larger and wider population. And then the clinical trials are also used to establish the efficacy and effectiveness of the vaccine in the real world, and then generate the clinical data for marketing authorization of the vaccine. So this is a simple slide I put together on the phases of the clinical trials. The phase 1 is typically a safety study where the sample size is between 20 to 50 subjects. Usually we do it in healthy adults, and it will be an open label, non-randomized studies, and it usually takes few months for us to complete the studies. Then from phase 1 we move on to phase 2, which we again determine the safety, immunogenicity, and the dosing range. Sometimes we take different dose levels, and try to see which is giving the best safety profile and also immunogenic profile. And it is again done in subset of target population. We would sometimes we expose the vaccine to the target population intended to the vaccine to be used. And again, we try to prefer, because it's a safety is the primary objective. For secondary, we trying to keep to an open label study with a single blind, observer blind studies are done. And again, in this, we try to introduce a placebo or a licensed comparator to compare our safety profile and also immunogenic profile to have a better idea on how the vaccine behaves during the phase 3. Then eventually phase 3, which are called as pivotal trials. Generally, we do immunogenicity trials, and there's another thing called efficacy trials, which I'll talk a little bit in my coming slides. Generally, this phase 3 trials are done with thousands of patients. It ranges and depends upon the kind of vaccine you're using. It might start from 2002, it might end up to 3,000-4,000, depends on how much safety data you want to generate. Because most of the vaccines are targeted for WHO pre-qualification. That's where the wider where we use. So that requires around 3,000 subjects safety database, which we try to generate in phase 3. And usually it is done on randomized double blind to avoid any biases. And preferably we'll use the licensed comparator. Placebo is less used until there is no comparator. And placebo is more used in efficacy studies. And it depends on the duration, depends from 1 year to 5 years is the amount of follow-up, how much safety data you want to generate, 6 months safety data, 1 year, 2 years, 3 years. For new vaccines, the more safety data you generate, the more it is good. So that is a bit about the different phases in clinical development. Then it's a very important question. How do we prove that vaccines work? It's just you inject somebody, and how do we prove that vaccines work? And if it's working, how much is the immune response? How is it in a measurable way, quantifiable way? That is what we want to do clinical trials with. So there are basically, there are different ways we approach the clinical trials, but the classical way is immunobridging studies, which are non-inferiority studies, which I'll talk in the next slide. And the second is an efficacy studies, are large scale studies where the subject size ranges from 20 to 30, 40,000, depending upon the disease, the prevalence of disease and the attack rates. And then there is an human challenge studies. These are the other kind of things where we try to prove, we try to measure the immune response faster. So what are these immunobridging studies? So assessing the appropriate immune response requires an immunoresponse markers. So the classic immunoresponse markers we use in vaccine clinical trials are geometric mean concentrations or titers. So this is the most commonly used measurement. This is done by ELISA assessment of antigens. The next is proportion of seroproduction rates. So there is something called correlate of protection. Correlate of protection is developed after several clinical trials, which tells us the amount of antibodies level or threshold of antibodies required for the subject, the person receiving the vaccine, to protect himself from the disease or infection. So that is correlate of protection. And for example, for pneumococcal conjugate vaccines, the correlate of protection after many clinical studies is established as 0.35 micrograms per microliter per ml. Sorry. And so when you vaccinate a subject and you measure his antibody concentration and it is more than 0.35, the subject is protected. So we use this concept to use it in a wider way in a clinical trials by using the proportion. So for example, in a clinical trial, it's a random number. We'll never have 100 subjects, sometimes for phase 2. So out of 100 subjects, if 97 subjects have antibody level of 0.35 microgram per ml, the seroproduction proportion is 97%. So these are the two main immune response markers. Then we have other things, 2,4, FOL4, the other parameters which we use, neutralizing antibodies, cellular immunity, those kind of immune parameters we use. So this happens in the classical immunobridging studies. This is what we do at a phase 3 level most of the times. Is we have a control group where we use a comparator, which is an approved vaccine which is being currently used in the market, and there's a test vaccine which is developed which needs the approval. So we measure the immune response markers, either the proportion of seroprotection or geometric mean concentrations or titers, or both we measure, and we do something called non-inferiority testing. It's a statistical method which is used to demonstrate the new vaccine is not inferior than the current vaccine. So it can be used. So I don't want to go into the details of the non-inferiority testing, the statistics involved. So basically we have a test group, control group. we measure the immune markers and then subject them to the testing of non-inferiority, and if it passes, we use this, the drug gets, the data gets submitted and we get approval. The next thing is the vaccine efficacy study. Vaccine efficacy is measured as a control in a controlled clinical trial based on how many people who got the vaccine developed any infection or anything what we call outcome of interest. It might be infection, hospitalization, of death, and it's compared with the subjects who have taken placebo for the same set of outcomes. So once the study is complete, the number of each groups are compared in order to calculate the vaccine efficacy, and it is basically the percentage of disease cases in vaccinated group compared to an unvaccinated group, to put in a simple sentence. So this is how it went forward. Can you go back a slide, please? Yeah. So this is how the vaccine efficacy studies are run. So you have a control group, you have a test group. A control group, we give the subjects placebo, whereas test group gives a test vaccine, the new vaccine which is developed. And then there is a set of, with the, on the attack rate and infection spread and the prevalence, there is a set rate of cases which drive the sample size also. And once the number of cases, infections are there, we open the study, and then we using this formula of which is present here, the vaccine efficacy. It is attack rate of unvaccinated people minus attack rate of vaccinated people, the rate by attack rate of unvaccinated people into 100 gives the vaccine efficacy. So yeah. So here is an hypothetical vaccine. I just put it for the presentation's sake. Before the study started, it is decided with its various parameters that when 261 cases arrive, the study will be unblinded. So there were 15,000 subjects who received the vaccine and 15,000 subjects who have received placebo. So one of the parameter is the infections within 2 months of the last dose, and then the hospitalization or infection within 2 months after last dose, or death after 2 months of the last dose. So these are three things which are monitored over a period of time. So in this case, based on the formula I provided in the previous slide, we have around 40 infections in the vaccine group and 200 in the placebo group. So when we take this and put it in the formula, the vaccine efficacy rate for preventing infection is 80%, and then there is hospitalization and 1 in the vaccine group and 19 in placebo group and 1 death. This is all together, when we put in the vaccine efficacy formula, shows the vaccine has 95% efficacy in preventing deaths and hospitalization. So I think this is what we extensively was used during the COVID, the pandemic, when they're determining the vaccine efficacy studies. And then there is, this is more interesting part, this is human challenge study, which we don't do it in India right now, but it's done a lot in England. The Oxford group does a lot of this thing. So human challenge study is where in a controlled environment, the healthy volunteers are exposed to pathogen and see how vaccine affects on that. So you have two sets of people. You have the test and the control. The test receives the vaccine, whereas the control group might receive a placebo or any other vaccine, but not intended for the disease, and they're exposed to pathogen. They're literally, given the disease, and then they're measured for their immune responses, and that is what is done in this human challenge studies. Usually, these human challenge studies are not taken for life-threatening or for the diseases where the medicines are not available. Very recently, classical human efficacy studies were done for typhoid, and there's now currently they're also planning to do a typhoid bivalent studies, which there's a very famous scientist in Oxford called Andrew Pollard. He does all this human challenge studies. So the advantage of this human challenge studies is the number of people exposed will be very less. The trials are faster, and there is and it helps in identifying the correlate of protection, which I was talking before, very fast. And this accelerates the vaccine development. This is a very radical way of doing a clinical trial, exposing people to a disease and then treating them with the pathogen, vaccinate them, expose them to pathogen, and see how it works. And then I'll quickly move to the safety part of the clinical trials, how we manage and monitor, and what is the things we watch out in clinical trials. So as I said, the safety is a vital in vaccines, as it's given to healthy individuals. We don't usually give vaccines to patients. It is given to healthy individuals, and especially infants, newborns, and children. So it is very safety becomes very critical here and very important. And in the clinical trials, we make sure that there is specific attention to serious adverse events and deaths if there are any. And we see the occurrence of the adverse events are monitored, and there's a compared to placebo and active comparator. And then the severity of the adverse events, how severe they are, mild, moderate, or severe, graded. And then the causality of the vaccine, whether it is related to the vaccine, it's not related to the vaccine, and if it is solicited, unsolicited, everything is done, monitored, and data is collected during the clinical trials. And how this is established during the clinical trials is we doing by safety assessment, and then we do something called post-marketing safety monitoring. So during clinical trials, what happens is, I'm talking of a classic vaccine trial. Once the subject gets vaccinated, the subject is kept at the site and observed for 60 minutes for any AEs or any other reactions. And the participants are given a diary for 7 days where they can document all the safety events if they're happening. And then it's make sure that all the adverse events are collected, both the solicited and unsolicited, and then there is a thorough follow-up of adverse events when the subject comes back to site for the follow-up visits, for the second doses or safety follow-up visits. There will be the investigator, who is a physician, will ask and inquire the subject of the safety, well-being, any abnormalities is noticed, and those kind of things are monitored. And the trends of adverse events are often compared to a licensed comparator to see what exactly is causing this. How can this be attributed to the vaccine? And then all the vaccine clinical trials are monitored by something called Independent Drug Safety Monitoring Board. So we have been establishing this. It has been one of the requirements which regulators ask us for most of the all of the studies. It has become a normal thing. So it is an independent body, not connected to company, which has physicians who are very well experienced in the vaccine informations and vaccine studies, and physicians, and then you have a lawyers just to see, to understand the non-scientific elements, and you have a statistician who are all not related or not nowhere connected to the company. And that's if the study is blinded, this DSMB are shown the unblinded data, so they know the safety events as per the placebo or the control group or the test. And they assess the safety in a very unbiased manner, and they then give a go ahead with the study, or terminate the study or stop the study or make any changes. So the safety is governed by them. And this is post-marketing safety monitoring. I think the follow-up presentation where Dr. Sridhar talks, I think he'll go more deep into that. But what we do is, post the clinical trials, once we get the marketing authorization, we still, through pharmacovigilance department, pharmacovigilance teams, continue to do the safety monitoring by active surveillance, by having sentinel sites. Sentinel sites is a concept of where we established the high rate vaccination centers, and we collect the safety information from there, and then drug events monitoring through registries and the electronic health record monitorings. And then there's a passive surveillance which happens. We all the vaccine companies have a mail address which is printed on the website and also on the package insert for the patients or the health administrators or the physicians to contact us if there are any safety issues. There's a 24/7 toll-free number and which is present on our the sponsor website. And even in the company website, we have a provision to report adverse events arising due to vaccine. And then we do something called spontaneous reporting through literature review, social media, internet, and periodic safety updates and signal detection activities. I think in your next presentation, Dr. Sridhar will talk more about that. So I'll talk quickly about Corbevax. This is a vaccine which we all worked during the pandemic, and it is India's first approved heterologous booster vaccine also, which is given for 18 years old. It is totally developed in India by all the scientists in India. It's it is developed in the company which I currently represent biologically. It is produced in East Expression System. It is an RBD subprotein vaccine, and it found to be safe and administered to children as young as 5 years and above. So we have data almost till 78 years age groups. So I don't want to dwell much into how we develop, what all these things. I just put two slides. One is the challenges what we faced developing this vaccine, and then how did we mitigate that. So one of the main challenges is, like I was talking of correlate of production few slides ago. So when pandemic came, the disease was new, there was no correlate of production, or there was something, which was not established at that point of time. Every company was trying to develop their vaccines, but there was no guidance towards where we are going. That was a very challenging time. And then when we started doing our phase 3 study, classically a lot of other companies which got their approvals in India or abroad had done the classical efficacy studies to prove that vaccine is working, the 30,000-40,000 subject studies. But by the time when we went into our phase 3, most of our population was exposed to COVID. We didn't find any use in doing that study because we would not, everybody had antibodies at that point of time. And then the other challenge was the disease was evolving. Every day there was a new drug which was refabricated and refurbished and started using for this study. So we didn't know where the disease was not. And then there were the variants. You have the variants coming in, and because the vaccine which we developed was for the Wuhan strain. But then came the other strains till the second wave and Omicron and all these things. So it was very difficult and challenge because we are not dealing with one variable here. And then when we are doing our immunobridging studies, it's paramount important that you give your vaccine to seronegative subjects, means subjects who have never been exposed to COVID. And it was very difficult. It was very difficult to find them because everyone had antibodies. Even they had asymptomatic, lot of people had COVID, they didn't know they had COVID. But when we went and when we were doing the studies, we found out that most of them were exposed to COVID. And then we had to conduct the clinical trials, which involves in working with the clinical sites and a lot of logistics during the lockdown, which was a very challenging for us because we were unable to move and lot of logistical issues. Then how did we mitigate this? First, I want to thank our regulators here who are sitting here representing here, Dr. Swati, Madam, Dr. Vishala, that they are given us an excellent support. They were hand holding us. They helped us in understanding, and they were there for us to any guidance, any challenges we faced. They were there with their good suggestions and good advices. And then they had done expedited reviews and given scientific guidance and facilitated for acceleration of clinical studies. So we had 11 SEC meetings. SEC is the subject expert committee meetings which approve the clinical trials, and we had 11 which were done at a very record time. And you won't believe, we used to have SEC meetings late in the evenings and it used to extend till night. That is how everybody worked during the COVID pandemic. And then we planned our clinical studies in a more proactive way. And then we needed around 4,800 subjects for all our studies from phase 1 to our phase 3s. To get that 4,800 subjects, we had to almost go to around 12,000 plus subjects. And we almost went to 90 plus clinical sites across India to get the subjects. So it was something which was very huge for us. and especially my team, the clinical staff, and the hospitals, they worked selflessly during the COVID phase to ensure there's a timely data deliverable, and so that we have data which we can present to the regulators to get the vaccine approved. And we used a lot of digital technology and remote monitoring during this phase, because the mobility was an issue. So we used a lot of this technology to review the data, and also then it took 14 months from phase 1 to when we got a license. I'll tell you how fast this was, because after that we got our pneumococcal conjugate vaccine, which got approved, and it took almost 6 years for us to go from phase 1 to get our marketing authorization. That's how things moved that time. And since March 22, the Corbevax has been used in young children from 12 to 14 years old, and almost 95 million doses have been given. So this is about what we did during the pandemic and COVID. And this is about our studies which we did. You can you can go and search in PubMed. We all the data was published in international journals, our phase 1s, phase 2s, phase 3s. This is about our studies that we have done. Okay, this is my last slide. So I just wanted to thank all the clinical study participants. Without them, we couldn't do any clinical trials. They do it very selflessly. They participate in the clinical trials. And research is not possible without help of those people who agree to take part in clinical trials. They help advance the science and giving hope to more promising tomorrow. So from all the scientific community who are working on the vaccines, we express a deep sense of appreciation and gratitude to all the study participants and volunteers. And without the investigators and clinical staff and the regulators, we can't do anything. They are the guiding force for us to develop our things. And again, sincere thanks to all of the frontline health workers who braved the disease during the pandemic and helped us in the eradication of pandemic. Thank you. Thank you very much.
Thank you so much, sir. We are really privileged to have you today with a great presentation on human vaccines. So, should we wait for another 5 minutes, sir?
Yeah.
To have some questions from the audience, if they have any. Audience, any questions?
If you don't have any questions, I think I have as a great presenter. And if you have any questions, I think my presentation was not. Yeah, please go ahead. Any questions? You can ask any question. Yeah, madam.
Sir, you are focusing on human challenge studies, especially done in UK. So, but that is really something of ethics concern, right? So, why that was very necessity for them to do that, and why not in India?
Madam will ask why not in India. Yeah.
Like Dr. Subhash has pointed out that controlled infection human studies would actually give you a greater insight, and during a normal time. That means you take a controlled set of population, you give small doses of the antigen to produce a disease and try to see whether my product, which is a vaccine, can prevent this disease. So this would actually help. These studies are really required. Second point would be like it would reduce the vaccine development timeline. That's what he very well presented in his speech or talk. So India too had thought of this, and today you must see the guidance document published by ICMR. There is already a guidance document in place. While you spoke, I was wanting to give you the exact text of this title of this document. It is called the Policy Statement for Ethical Conduct of Controlled Human Infection Studies in India. This is published in 2023. So please read this document. There was a lot of discussions, debate when this document, a draft document was published. People have contested the idea, and but we had the backing of the WHO guidance itself, and then India too framed its own policy. It's out there. You can read it, and you can find my name as well.
Thank you, ma'am.
Good evening, sir. I would like to focus a question on, although there are so many drug agencies which are involved in marketing any product like CDSCO or anything, why are many ADR still reported after post-marketing, sir?
See, that is what the post-marketing effects are there. You know, you try to capture the adverse events during clinical trials, and then a lot of times during post-marketing ADRs report. The challenge here is people don't tend to report ADRs. So well, we follow up with the sites, and when we clinical trials we educate them, but in real practice, real world, people usually don't report ADRs. So kind of ADRs we receive. Madam has to say something.
Dr. Thuluva, it's the other way around.
Yeah.
You don't report serious adverse events or in clinical trial, it's a crime, like Dr. Prabha.
Correct.
So all serious adverse events in clinical trials have to be reported to CDSO.
Yes.
Happening in a clinical trial.
Yes, ma'am.
Your question was if the product.
Why
Well, our question was why there are more ADRs post marketing.
Yes.
So, answering that. So, in clinical trial, the population that undergo the trial is very uniform set of people and very limited. In vaccine, you can see there are around 15,000 people who had to be screened to do a trial in 5,000 people, right? In clinical trial, very controlled studies and controlled settings with specific inclusion-exclusion criteria. The idea of identifying those chance ADRs in clinical trial is possible. That means if you screen 10,000 population, one serious adverse event maybe you can detect.
Yes, ma'am.
So even if you did a trial of 10,000 population to detect one ADR, I mean, you are blocking the pathway of the drug approval to be market access. So the idea is to do broadly post-market surveillance, and it's not that we can understand how a human behaves. The human itself is a complex
Structure.
Human. I mean, I should say object, or I don't know, it's a living being rather. Very difficult to understand. Various serotypes, various genotypes, various changes happening dynamically, environmental changes, etc. So today we can predict everything with 10,000 population, and then you can scale it up to multiples and billions of population. That would be really interesting, and we should always look for that. It's a very good question, but phase 4 monitoring of the patient safety for long-term effects, as well as capturing the effects which could not be identified during a clinical trial in
Maximum
limited
poll.
population.
Yeah. But ma'am, being subject oriented people, we can understand about it. But layman, with one ADR report which is being mouth propaganda, with the mouth propaganda, they will stop taking. I know so many people who have not taken vaccine, COVID vaccine as well. Just thinking about like ADRs that others have reported, and even till today. So many who have already taken the vaccine also report so many adverse drug reactions which are unanswered, and there is no one who is answering about it. So that is
There is a difference between serious adverse event and ADR. To say this is an ADR and this is the causative reason, that is the vaccine is the causative reason, that itself is a huge science.
Okay.
That's causality assessment.
Thank you.
So before saying it is an ADR, first we should be clear on our mind whether it is related to the drug, or is it coincidental.
Yeah.
So vaccine hesitancy was there, but ultimately as pharmacists, as scientists,
We should educate.
we should educate the population that you get more protection as compared to having no protection without a vaccine.
Thank you, ma'am.
I think the next presentation, which Dr. Sridhar speaks a lot about the ADRs. Okay, Dr. Ramkishan.
With permission from the chair, very interesting. Vishala madam already explained, madam. You are from academia side or industry? Academia. Good question. So why we are having more ADRs after approval of market authorization? That is your main question, right? So in our country, what is the population, madam? As in yesterday midnight, 143 crores, 78 lakhs, 55,000, 232. Abhi kitna hua ban pata nahi, right? So when we go for the distribution of the product, so regulatory setting already madam told control setting. So now because limited people are exposed during the clinical trial process. So once product is approved, distributed to various market, maybe domestic or international commerce, so a lot of exposure, right? So then again, the main challenge what we are facing currently is not only in India, globally, people are not having that preparedness. In our country, what is the shortage of our total doctors? How many people really taking the active surveillance and passive surveillance? Doctors are round the clock busy with their own clinical practices, right? And paramedical nursing people and technicians are filling up the what we call the ADR form. So now things are changing. Government of India published the guidelines. Now it is a law, mandatory requirement with respect to submission of ADR reporting form, and we are having analysis and ADR forms, and also we are having app with respect to Indian Pharmaceutical Commission at Rajnagar, Ghaziabad taking care, and we are sensitizing. With respect to vaccines, it is different, not ADR. Adverse events following immunization. AFAs are different from the ADRs. So, madam did a lot of work. From the regulatory side, we are putting efforts. We need support. Each and every company should have a well-defined pharmacovigilance system in place. आज law बनाया. We are putting efforts. At the same time, we are creating awareness through IPC. I would like to congratulate all the chairman, co-chairman, and panelists, Mahendra Bhai Patel, and all the people sitting here. We are educating people, and we are also having close access. WHO also assessed the Indian Pharmacopoeia Commission with respect to pharmacovigilance, and we are also having one of the major indicator in national regulatory assessment by the WHO Geneva people. They will also come and do the assessment. So putting efforts globally, we are having name, and we are having more than 300 ADR centers in our country, and taking care of the more than 2 lakh people. ADRs are this thing reported. We are also having separate well-defined casualty committee with respect to analysis in case of any serious adverse event or death is happened, including a small injury also. We are having committee consisting of all medical experts and taking lead. And first time in our country, we have incorporated the compensation formula in our regulation. Nowhere in the globe we are having that of this thing. So credit goes to Drug Control General India and Ministry of Health and Family Welfare Department. So putting efforts, educating people, ultimately patient safety is very, very important. Your mom, your daughter, my daughter takes, how do we react? So these are all the things. Thank you very much. And Honorable PM Sahib is taking, and Government of India is going in a different direction. We are going to be number one by 2030. Thank you very much.
Thank you so much, sir.
Good evening, sir. Sir, now we have, after COVID, we have seen more vaccinations more towards COVID and all. So, in case of Moderna vaccines and Covishield, we have seen a lot of side effects, like they're suddenly popped up, maybe deaths and all. So, in case of Corbevax, how confident the safety of your vaccine, and how you are going to address the safety issues?
See, we have already administered around 9.5 crores of vaccine, and we didn't find any life-threatening or any big thing other than our solicited and adverse events which we found in clinical trials. There were some AEFIs which are reported, and we thoroughly investigated them. We work with the CIDISCO group with our PSURs and periodic updation of this thing. So overall, I can say the Corbevax vaccine is very safe when compared to other vaccines outside India, which you're talking most of them. Corbevax was very safe.
Thank you, sir.